The lecture is about reviewing the family problem case. The case is about story about single gene inherited familial disorders and how it progressed from the molecule until impacting the family and the society. So the full case should be understood under this paradigm. Paradigms mean a single gene inherited problem might mount up to affecting the whole family's life and the whole society. Now you have 8 key questions you should answer during this case. First key questions that you should have studied during the week. You'll go to revise these questions, revise your learning, but I will not discuss in details every aspect of the. First question will be, where do single gene abnormalities come from? How can gene abnormality change cellular functions? How would these cell changes be reflected on tissue and organs, consequently resulting in a disease? And what are the normal structure and function of the tissues and organs affected by the disease? Next questions will be, how does the understanding of the molecular changes help in choosing appropriate diagnostic tests and therapies? How can environment interact with the resulting genetic changes? And what is the role of healthcare services in genetic disease? The 8th questions is related to behavior and social sciences, and it's mainly how this line of disease or pathological changes will impact will result in a chronic disease that will impact the family and social relations, and how the family and the society should respond to such problems in the functional impact. Gene abnormalities comes from the central paradigms, deals with the DNA resulting in an RNA that is translated into protein. So proteins is the main outcome of this process, and the protein will function in different ways in the body. One of them is forming transport molecules, receptors, etc. So when the DNA sequence when they occur, the DNA is how the mutation happens. When the mutations, these are differences or changes in the DNA sequence, they occur, sometimes occur in the somatic cells, and these may result in a cancer and developments. They might occur in the germ cells, and there is a risk that offsprings will come structure and function disabilities, which means the inheritance of. But sometimes these changes happens de novo or new mutations in the germ cells, and later on become inherited, but it become de novo. So this is very nice illustrations of how mutations happens or the type of mutations. If you think of a gene like a sentence with a meaning, that should start by a coding of that and ends when the transcription mechanism find the end. So probably you would think if the sentence say the man saw the dog hit the can, and this should end the sentence here. End the sentence here. So point mutations mean that one base pair changes. So instead of the man saw the dog, it will be the dot. So the meaning would be different. The deletions mean deletion of the 3 codon, the triplet codon that results in the amino acid sequence, an amino acid molecule in a protein sequence. So this might result into a very truncated or a very meaningless sentence, like the man saw that, and then hit the can. Insertions mean that we insert a codon, and meaning that we insert a full amino acid in the sequence. So the man saw the fat dog in the can, hit the can, and so insertion again change the meaning of the sentence. Frame shifts mean that there is one deletion happen, so the triplet coding will change until the dog D is omitted. So the transcription starts reading from OGH-EITT, so it misses the end, and this results into a very new protein. Sometimes non-production of the protein. In cystic fibrosis, if you want to translate what you have here now into the cystic fibrosis gene, so cystic fibrosis primary caused by mutation of a single gene. This is the gene, the single gene is called cystic fibrosis transmembrane conduction regulator, CFTR. It's located in chromosome 7. The protein products of the gene acts as a channel connecting the inner part and the outer part of the cell. So it connects the fluid. So it mainly affect or responsible for the movement of chloride across the cell wall. So this is the base pair deletion is mostly delta F508, mean that deletions mean delta. Delta means deletion. F is the for phenylalanine, and 508 is the sequence of this amino acid in the resulting protein chain, a protein, or in the resulting amino acid sequence. So the proteins is produced. It is processed to form these channels. If we have a deletion, so we have loss in the phenylalanine at 508 positions. So these are the 3 base pair deletion mutations. Okay, so these are the common inheritance patterns. I suppose you have studied this week. So this mutation is inherited as autosomal recessive pattern. You should be aware of, and should you go study the difference pattern of inheritance between autosomal dominant, autosomal recessive, X-linked recessive, X-linked dominance. As well, you should know the symbols we use in drawing these pattern, the inheritance pattern. Okay, these are international symbols you have to study and know during your work this week. Okay, another concept you went through, or you should have went through during the week, is the population genetics. What is meant by population genetics is the study of the distribution and changes of allele frequency in populations. Oh, so what does this means? What is the importance of this? Think about screening of tests for genetic diseases. So we know now that not a single problem, not a single genetic aberrations results in a disease. So some diseases like cystic fibrosis as well can result from several up to 1,300 mutation types. However, population genetics tells me that the delta F508 or 508 mutations happen in 70% of the cystic fibrosis cases. So when I go and I suspect a patient with cystic fibrosis, and I do this in the Western community, where I know that 70% of the mutation comes from this from some 70% of the cystic fibrosis cases have this mutation. So I go and study these mutations, this delta F508. Okay, so this is the importance of population genetics. Is this we know through it the frequency of the distributions of the different alleles or the different problems. While we know that, for example, in Iceland, 100% of the cases comes from this, but in a specific population, like in Pakistan, for example, you have another type of mutation called Y569D. So when you examine or when you test genetically for a child from Pakistan, and you are suspecting the cystic fibrosis disease, so you don't go and test for this mutation, you go and test for this mutation. Obviously, no one can test for all the 1,300 or more mutations that is implicated in the cystic fibrosis. So this is the meaning of population genetics. Again, population genetics mean the study of the distributions and changes of allele frequency in populations. Why this is important? It's very important when we think about screening populations, when we think about studying the origin of diseases in certain population. It might be one disease, but it originated from different mutations in different populations. So this is a very good example for you to know. In the UK, for example, when we test for cystic fibrosis, automatically we go for the or they go for the delta F508 mutations. While if it is a person coming from Jewish populations, it will be that mutations. In the Pakistani population, it will be that mutation. And this is very important for you to understand. So the second questions you should have answered during your study that week is how can gene abnormalities change cellular function? So we understood now where the gene abnormalities comes from. We understand central paradigms, DNA transcripted to RNA translated into proteins, and the protein is processed to do the functions. And we know now that a change in the DNA will result in a change in the protein sequence. And we know when you should have by now know where the mutations come from, their types. Now, the second questions will be how can gene abnormality change cellular functions? Okay, so either these new proteins or this problem that resulted from the translation into that protein, either the cell will gain the function or lose the functions. So like here, when we lose the function of the cystic fibrosis, so the cell loses the function of regulating chloride across the membrane. Or there is a novel property of the protein synthesized. So some proteins, the new resulting mutated protein or mutated DNA that resulted in a different protein results in a novel property, another property of the protein. Very good example, the sickle cell anemia, anemia Mengele. In sickle cell anemia, the resulting hemoglobin has a very different property than the normal property. Now, neutral or there's silent mutation, so no harm or benefits. It might happen, mutation might happen, but the resulting protein is neutral. It gives the same function. So there is no harm or benefit happens. Sometimes there is abnormal expression of the gene that results into activating a gene, and these activations results in a disease. The very famous thing is the cancer causing genes, the oncogenes. The oncogenes, the theory says that the oncogenes should have stopped working when that person reached maturity or full growth. But the theories say when something happens that results into activation of the oncogene, cancers results. Okay, so what happens in the receptor proteins? The membrane-based protein, which is a channel that is found in the plasma membrane, normal epithelial cells. It's a single polypeptide chains of 1,480 amino acids, and it is a cyclic AMP regulated chloride channel. So this means it needs energy to move the chloride from inside the cells to outside the cells. When it moves the chlorides, it takes with it sodium. And when the sodium is taken or moves, the sodium usually moves water with it. This channel might be positioned in a way that it results into moving chloride from inside to the outside. But also in certain tissues like the sweat glands, it is positioned in the opposite way. So it moves chloride from outside to the inside. And so it absorbs water rather than secreting water. Okay, so the classes of the CFTR mutations, depending on the type of mutations, you might have a defective productions from the start, might have a defective processing, and this is what happens in the delta F508 mutation. So the protein is processed, is the chain, the amino acid chain is produced, but it is not processed. It does not take this complex picture, cannot take these processing pictures. And this processing picture, or this three-dimensional orientation of the protein is what forms these channels. So these channels need this processing issue. Another mutation might result in defective regulation, so the protein does not reach the surface, or defective conductions, so it reaches the surface, but the function of the channels is defective, meaning that it does not secrete chloride. It might take the chloride in. It might reduce in the amount. So protein is produced, but in a low amount. The classical delta F508 results in a defective processing, so it does not reach the cell membrane because the processing happens before the reticular endothelium inside the cells. So the CFTR protein results into epithelial function problems. So epithelial containing CFTR protein exhibit an array of normal functions based on the type of epithelial cells. So these cells, the epithelial cells, occurs in different organs. So epithelial cells, epithelial line, epithelial tissue happens in different organs. Depending on the organs, these channels will function differently. So for example, it maintains in the mucus, if it's epithelial, the mucus layer in the airway, its main function is to maintain the fluidity. Main function is to secrete or excrete enough water to the surface of the airway. So it results into decreasing viscosity of the mucus. So the other things that the volume absorption, if it's in distal intestines, function a different way, function towards absorbing the water and the nutrient content from the lumen of the intestine into the cells, into the intestinal mucosal. In the sweat ducts, it functions to absorb the salt without absorbing the volume, meaning that I don't want in the sweat a lot of sodium excreted, because if there is a lot of sodium excreted in the sweat, the person will be at the risk of hyponatremia, meaning that low sodium in the body. Low sodium in the body results in several problems. So in the sweat glands, the same channel, the same protein in the epithelial cells act to absorb sodium without absorbing much water. Okay, in the proximal intestine and pancreas, its main function is to secrete the pancreatic secretions or the pancreatic enzyme. So the volume secretory function. So the dysfunction of this protein resulting from a dysfunction of the gene results in a different effects depending on the system or the tissue, the organ that they are functioning in, they are working. So how would the cell change be reflected in tissue and organ, consequently resulting in a cell? So the main paradigm, the main thing that you have to understand this week, that we had one gene problem that resulted in one protein problem. This protein problem is present or important for one cell type, but this one cell type is present in several organs. So when we translate this, so CFTR gene, it produces the CFTR protein. CFTR protein is very important for the epithelial cell lines to work, but epithelium as a tissue, as a cell type or a tissue type of a tissue is present in different organs. Present in the respiratory system, the airway, present in the GI, small intestine, pancreas, biliary tree, present in the sweat glands, present in the vas deferens, which is a male internal genetic 6 organs. So because it is one tissue, but it's present in different organs, each organ should function in a certain way. And because of loss of function, the organ is affected in a different way than the other. Now you should understand that you need to, when we go through this, the function in each organ, in the lung, as I said, there is a high rate of sodium absorption and low rate of chloride secretion, reducing salt and water content in the mucus. So it depletes what we call periciliary liquids. Okay, so this is the effect, the pathology that happens. The mucus will adhere to the airway surface, leading to decreased mucus coherence. So the person will have plugs of mucus. These plugs of mucus will invite infection, will invite Staphylococcus and Pseudomonas infection. The gastrointestinal tracts in the pancreas, as I said, it should promote secretion of pancreatic enzymes, but because it is not there, there is limited function of chloride bicarbonate exchanger to secrete bicarbonate. It leads to retention of the enzyme in the pancreas. And these enzymes are very powerful. It ends into destructing the pancreatic tissue. In the intestine, in the terminal ileum and cecum and ascending colon, the decrease in water secretion leads to thickening mucus and specific intra-ileomal content. And this will result in the obstruction of small and large intestine due to accumulation of the sticky mucus and fecal material. This happens in the early life, in the mucous membrane, in the newborns, in the intestine of the new. The gastrointestinal tracts in the biliary tree, it results into retention of the biliary secretion. So biliary acids, bile acids, that is very important to be secreted from the liver, from the biliary channels to the small intestine, that will help into digesting lipids, is not secreted, it's retained. So what happens? The livers get injury. So we call biliary cirrhosis. Cirrhosis means تليف الكبد, so biliary cirrhosis. And the bile duct result bile duct perforations, chronic infection, chronic cholecystitis, chronic cholelithasis, which is the stones in the gallbladder. In the sweat glands, it results into inability to reabsorb sodium chloride back into the body. So the sodium chloride, the sodium and chloride will pass in the sweat through the sweat ducts, and this will result into loss of NCl or sodium chloride in the sweat, and hence water will be lost. So this will result in a when you think about the magnitude, the number of tissues, the number of organs affected, you can imagine what will be the effect, the pathological effect, the disease effects. So we have a long list of complications at the clinical level. Obviously, malnutrition due to malabsorption, intestinal enzymes, pancreatic enzymes, all of these things are not there. So there is malnutrition, malabsorption. Bronchiectasis, which means destruction of the bronchioles, pancreatitis, which means inflammation of the pancreas, diabetes mellitus, I think you know this, cirrhosis, talayuf, osteoporosis, renal enzyme, allergic bronchopulmonary aspergillosis. Aspergillus is a fungus that is invited to infect the lungs, but it results into allergic reactions. So plus infection, we have some sort of allergic reaction due to this fungal infection. Liver diseases with portal hypertension, chronic respiratory failure, acute respiratory failure, delayed puberty, and short statures. These are very concise list actually of the complication that might happen due to cystic fibrosis. And if you can see it, it's affecting very wide range of organs. Now, what is the normal structure and function of the tissues and organs affected by the disease? I will not go through this, but this is a reminder for you that this week involves that you have to study the respiratory system anatomy, cystology. You have to all the physiological issues like cough reflex, transport across the epithelium, mucociliary clearance, which I will discuss in details in the next few slides, epithelial lining, conducting versus respiratory zones, upper and lower respiratory. Now, what's mucociliary clearance? Mucociliary clearance is the way the airways clear the mucus, clear the pathogens that enters with the respirations. So there is what we call mucociliary apparatus, which is an important primary innate defense mechanism, innate primary defense mechanism against the infection of the airway. While we take breath, a lot of organisms, a lot of particles enters with the respirations, and without the ability of the conducting airway to remove these organisms, these particles will stagnate inside the lung, resulting into infections, resulting into obstructions, resulting into a lot of problems. So the main apparatus responsible for this, or the main mechanism, called mucociliary clearance, and it became clear in the last 20 years that is behind it what we call mucociliary apparatus. So it protects the lungs from deleterious effects of inhaled pollutant, allergens, pathogens, mucociliary dysfunctions. Mucociliary dysfunction is a common feature of chronic airway disease. So what is this apparatus formed from? It forms from cilia, which is very fine organelles that protrude above the cell, these lines, these very fine organs. And these cilia is bathing or swimming in what we call airway surface liquid layer, which is called the preciliary liquids. Over this liquid, the mucus is floating. So the mucus, the goblet cell that produce mucus, it's producing the mucus, but in order to prevent this mucus from sticking on the surface of the cells, it flows. These preciliary liquids functions to carry this mucus above the fluid level. So the mucociliary, the cilia, when the cilia stroke towards this side or toward that side, it moves the fluid easily, and above the fluid, the mucus moves. So what happens usually is that when you cough, this ciliary apparatus helps you to dislodge the mucus from your airway and spit it out. So this is a protective layer, and this is the normal functions of this. So in this mucus, the mucus will function as a sticky material that will catch the organism, catch the particles, and hold it until you cough, or until this cilia moves this mucus, or through moving this fluid layer to the outside. Okay, so there's another picture. So we have a cilia, we have a solution layer, water layer, and above the water layer, there is this gel formed by the mucus. The gel traps the particles, and The fluid helps to make this gel float, not sticking on the surface of the cells, and the cilia will move easily through this solution layer to stroke outside, to move this layer outside of the airway. So what happens when there is a problem with this mucociliary layer? So as I said, when you have a problem, the CFTR problem, then the amount of fluid, amount of this periciliary fluid decreases because there is no water secreted. So the cilia here become exposed to the mucus and it becomes destructed and the mucus becomes very near to the surface area. So if you imagine this is the normal histology, this is what happens when there is abnormality. The cilia, these fine lines are not there, destructed, and these large particles of mucus are sticking to the surface of the epithelium. Okay, so this is the mucociliary apparatus, which is part of the physiological, actually the histological, anatomical, and also you have to understand the physiological functions and how this physiological function is affected when you have cystic fibrosis. Okay, there are other related topics you have to study during the week: GIT structure, the exocrine pancreas, physiology of digestion and internal absorptions. I will not go through them. You have to know, just a reminder, you should by now know exactly what are the enzymes that is involved in digestion, starting from the salivary gland enzyme, salivary amylase, what happens in the stomach, what happens in the pancreas. All these enzymes are part of your studying this week. Okay, so what happens? What is the problems when these are affected, when the GIT affected? There are another issue. I think it's mentioned in the text of the case that the child, despite having no abnormal cognitive level, no IQ normal of this baby, of the child in the case, and the problem you have, despite this, he had a very low school performance. And the issue of the question you should ask yourself, why? Why as a person who has normal IQ, you expect to have a normal cognitive functions, have a problem in studying or a problem in socialing? So this comes when you understand the normal cognitive development of children. So there are two theories you are responsible for this week, which is the Erikson and Piaget theory of cognitive development. Why the person developed, how the understanding of a human being developed from since birth up to becoming an adult or a functioning person. So this, in a simple way, I will not go through them in details. In a simple way, the theories of development says that for you to develop from a level to another level of cognitive, for example, if you want to understand what's meant by abstracting, the highest level of operations, you need to go through certain challenges. So the person in the first 2 years, the challenge is sensory. The challenge is that he can master, for example, suckling or oral feeding. And as he goes up, a person has to be subjective to certain challenge in order to develop certain cognitive abilities. One of these challenges is challenging of dealing with other person, is playing with other persons. If we prevent a child from contacting any other child, or decreasing this contact, or preventing the play of a child, the child might have a normal IQ, but he will not be able to develop cognitive abilities. He will not be able to study. He will not be able to make logical connections. Okay, so again, go and read about Erikson and Piaget. Nutritional requirements. Again, you have to know the nutrition requirement for children's major nutrition requirements. What are we need for carbohydrate in the first 12 months of life, fat, protein, calcium, iron, zinc. All of these are revision of what you should have read this week. Okay, this fifth question is, how does understanding of the molecular changes help you in choosing the appropriate diagnostic test and therapies? If we know that there is a problem happened that way, how it will help us to diagnose, how it will help, what should I test in the patient to diagnose the disease. So the first thing you learned this week about diagnosing such patient is sweat test. And sweat test is mainly resting on understanding that in cystic fibrosis patients, the cystic fibrosis patient is not able to absorb all the sodium from the sweat as a normal patient. So if I could measure the sodium in a sweat, so probably I might be able to identify those patients. So if I found that sodium and chloride content is higher than normal, so this might be a clue or might be an indicator to diagnose cystic fibrosis. And this is the basis actually of sweat test. So it's a very good sweat test, is a very good reflection of ion transport for the whole body. Actually, it's non-invasive. One of the things that's good for sweat test is non-invasive. I don't need to take blood sample. I don't need to do endoscopy. I don't need to open the patient or take a biopsy, for example. It's very reliable, okay, if done probably, and if adequate weight of sweat collected in diagnostic test. And this is the mainstay for, you have to understand where sweat tests come from. So you might get a questions, why I'm using sweat test? What are the pathophysiological basis of using sweat test in diagnosing cystic fibrosis? This is an exam question for you. Okay, why? And how sweat test will help me, and what is the basis, what is the pathophysiological basis of using this test in diagnosing cystic fibrosis patients. The other test is mutation detection and depends on population screening. Again, here comes the population genetics. So in every population, you expect to have a different pattern of inheritance or a pattern of mutation. So you go and test for that mutation. So you need to have knowledge about population genetics of cystic fibrosis. Okay, this is again, I hope you should have, I think you should have taken in the histology how sweat is produced, but I'm going to revise it with you. Usually in the derms, we have the sweat glands, and as the duct moves out up to the derms, up to the epiderms, we have the duct. So in the sweat gland, there is a secretion stage. So water, sodium and chloride will enter to the sweat glands passively, taking with them the water. So a lot of water and sodium chlorides are inside the sweat gland. As the ducts moves up, transporting this fluid, this unrefined sweat, a refinement process should happen. Otherwise, if the body loses all the fluid here to the surface, body will enter into massive loss of fluid, dehydration, for example, and massive loss of salt. So what happens is that if we have a high flow, meaning that there is a lot of heat, so sodium and chloride will be absorbed. Also, if we don't have a heat, sodium and chloride will be absorbed more. And as we absorb sodium and chloride, some water will be absorbed. So at the end, what remains in the sweat on the surface is a salty solution that is relatively fine-tuned concentration of sodium and chloride. So there is no sodium and chloride loss, a lot of sodium chloride loss, and there is acceptable amount of water loss. What we want in the sweat is the water, because the water, when the water evaporates, it decreases the body temperature, and as it decrease the body temperature and helps in controlling body heat. Body heat is controlled through sweating, one of the main mechanisms. But the sweat, what we need in the sweat to control the body heat is the water. So we want the water to be secreted, but physiologically, water does not move without moving sodium and chloride. Okay, there is no way that there is a selective water excretion. Water is excreted passively when we move sodium and chloride inside there. But as we secrete this or excrete the sweat, we don't want to lose a lot of sodium and chloride. Okay, so the body selectively through the CFTR channel selectively absorbs sodium and chloride and leave the water or much of the water up. So some sodium and chloride will come to the surface, most of them will be reabsorbed, and a lot of water will come to the surface, and little water will be reabsorbed. So in cystic fibrosis, what happens? There is no refinement process. So what comes here is lost on the surface. So what can you say? What can you imagine happens? That person will have a lot of sodium in the sweat. So this is the basis of the sweat test. I measure the sodium and chloride. I measure the chloride and sodium. Okay? And the person will lose a lot of water as well. So these persons are liable to dehydration in hot weather. This is very important. So other tests probably you come across during studying this case, the trypsinogen assay. Trypsin was one of the enzymes secreted by the pancreas, and the precursor of this is called trypsinogen. The trypsinogen is the inactive form of trypsin that is stored in the pancreas. So what happens when the pancreas could not excrete this trypsinogen? This trypsinogen will accumulate and then reabsorb it into the blood. So when we measure trypsinogen in the blood, this might give you a clue that there is some dysfunction in the pancreas. And I know that dysfunction of pancreas secretions can come from cystic fibrosis. So it's a screening test. It does not. It is not a confirmatory test. So we used to screen in what we call Guthrie test in the newborn. Guthrie test is the screening that happens to every newly born child. Every newly born child, we take few drops of blood from the heel, put it on a paper called Guthrie paper, and this paper goes to do a lot of testing on inherited diseases. One of these testing is the immunoreactive trypsinogen assay. If we find it high, we bring this patient or this child back to do confirmatory test of cystic fibrosis. Okay. It is not diagnostic because in heterozygous patients, carriers of cystic fibrosis, it's also high. So it is not confirmatory of a clear-cut case of cystic fibrosis. Just give me a hint. Just give me a suspicion. So it is a screening test. So fecal fat, if you do not have enough digestion, the fecal fat increase. Again, it's an indirect indicator of fat digestion and hence pancreatic enzymes. Lung imaging, X-ray, lung functions, pancreatic function, liver functions. All of these are tests used in those patients. But you need, again, for your level, it's not about memorizing these tests or the values. It's about explaining why they are useful. So the question line that you might face in your exams is why fecal fat is useful in screening for the cystic fibrosis. So you go from the start. You explain cystic fibrosis protein, you explain its function in the distal intestine, in the proximal intestine, in the pancreas, and then you deduct that there is no enzyme secreted. So no enzyme secreted, there is no digestion. So there is no digestion. So there is excessive fat loss in the stool. So this is what's meant here. So the testing here is not about, or the tests we discuss here is not about the tests themselves or their values or their use in the clinical scenarios. It's about you explaining why these tests are important or are functioning in this disease. So how are we going to treat again? If you understand the pathology, you might think, okay, we have excess mucus in the airway, so we need to use the drug called mucolytics, drugs that liquefy the mucus so the person can easily spit out the mucus. We can manage and prevent pulmonary infection because we know that pulmonary infections are invited. The GI and the nutrition, pancreatic enzymes, we give pancreatic enzyme. Of course, we have a feeding problem, so we need to increase the calories, increase the protein. The baby is weak, so probably we need to force him to eat. So we use things like nasogastric tube or opening a hole in the stomach. We call gastrostomy feeding tube. Okay. How can environment interact with the resulting genetic changes? This is easy because we have problem here with the expulsion of the harmful material and particles. These invite infections. So mucus clogs in the airway invite infections. And this is an X-ray of severely damaged lung due to infections. And it's easy to why there is a respiratory infection. So why? Because there is poor excretory or poor cleaning mechanism of the airway. So the mucus is sticky. The mucus clogs invites the adherence of the organism to the surface, invites the adherence of harmful particles to the surface. Okay, the respiratory infections you should know this week is the RSV, respiratory syncytial virus, chickenpox, Haemophilus influenza, Staphylo aureus, Pseudomonas aeruginosa, Prochylidia specia, and non-tuberculous mycobacteria. These are the organisms that mostly affect those patients, and these are the organisms that you have or should cover this week in the microbiome. Okay, it's 9 o'clock. I will stop here. I stop here the lecture because I have other commitments.